All right, looks like we're ready to go. My name is Caleb Calhoun, and I'm going to be going through lesson 11 practice problems dealing with translation. So for the first question, we're asked about translation in E. coli, and we're dividing the three different stages of translation. We have initiation, elongation, and termination. So for part A, we're asked what mRNA sequences, rRNA molecules, and tRNA molecules are involved in translation initiation in E. coli. So the mRNA sequences that are involved in initiation are the Shine-Delgarno sequence. And what that is upstream of where translation starts, there's a sequence of these nucleotides, and they determine where the ribosome, the 16S rRNA, binds. There's also a start codon. It's the 5-AUG start codon. You can see it up here. And that is where translation will start occurring. That's where the polypeptide chain will start being formed. rRNA molecules, there's the 16S rRNA, which is a subunit within the ribosome. And within this ribosome, that subunit is where the Shine-Delgarno sequence comes in, binds onto. And so that way it's ready for when the AUG start codon comes. Then there's the tRNA molecules, and the initiator, the tRNA molecule that initiates translation is the one that will bind to the anticodon, will bind to the AUG codon. Initiation says what proteins are involved in initiation. There's the IF3 and the IF2. And these are involved in helping the 16S rRNA and Shine-Delgarno sequence bind. So you have your Shine-Delgarno sequence and then the 16S rRNA. And IF3 initiation factor is what helps with that. IF2, this helps the tRNA to come in and bind to the AUG start codon. So both those initiation factors are proteins. The mRNA sequences and rRNA molecules involved in translation. So each codon involved in translation there's three of them. There's three bases. Sorry, I'm forgetting the words. There's three bases, and each of the three bases correlate with a different tRNA molecule that has an amino acid on it. And so these are the mRNA molecules, I guess, or sequences that help in elongation. And then as far as an rRNA molecule, the 16S rRNA, that helps in proofreading and making sure that there's no mistakes. The 23S rRNA, this is the rRNA or the ribosomal RNA that actually helps synthesize the polypeptide bond that is occurring as the mRNA goes along the ribosome. There's a polypeptide chain that's formed, and 23S rRNA is what's making that happen. Proteins involved, there's the EF2 and then EFG proteins. EF2 helps deliver the tRNA molecule to the A site of the ribosome. A site's over here. And so the EF2, it consumes a GTP molecule in order to make that process happen, the tRNA to come down. And then the EFG helps translocation. So it helps the tRNA move from the A site to the P site. So go from here to here. mRNA sequences and proteins involved in translation termination. So there's a stop codon, and it's one of, I think, three different stop codons. One of the examples is UAA. So that when translation is taking place, UAA, when it enters the A site, it helps recognize that that's where translation needs to stop or terminate. The proteins, you have two release factors, release factor 1 and release factor 2, and those just recognize difference of these sequences, and they help the subunits to disconnect and the polypeptide chain to leave the ribosome. Okay, question 2. This concerns the hydrogen bonds formed between mRNA codons and tRNA codons, and with a wobble rule. And so if the mRNA codon has the sequence UUU, the sequences that the tRNA anticodons can then recognize are, so I'll write this out. Yeah, you can see. So if you have UUU, the wobble rule states that these two have to be matched up perfectly, these first two bases. Here, I'll draw 5 prime, 3 prime, 5 prime. So these two bases have to line up perfectly, but here there's wobble room. And so the different bases that could be there in this molecule are G, A, U, and I, which is that's specific to tRNA molecules. So those are the different sequences that can bind for UUU. And then I'll go through these other ones kind of quick. GCG, the tRNA codons that can recognize GCG. So obviously the first two, they'd have to be C and G. So each of the 3 different ones have to have C and G. But then the third one, it can be either C, A, or U. And then we're asked for IAA. And for that one, the first two need to be U. So U and U need to match with the I and A. But then there's the wobble rule. And so it could be either U, C, or A that can bind to that last one, or that can be on that last one. And then with AUC, first two have to be G and A to bind properly. But then the last sequence, it can be U, it can be C, or it can be G. Hopefully that makes sense. Question 3. Okay, this one's kind of a long one, so I drew it out. We have this certain sequence right here, these nitrogenous bases. And this is an mRNA molecule. For A, we're supposed to identify the binding site, the start codon, and the stop codon. So the binding site is right here. This is the Shine-Delgarno sequence right there. The start codon is always AUG. In E. coli, it's mostly AUG. And then the stop codon is over here. We see UAA. We're asked, what is the amino sequence of the polypeptide encoded by this short gene? So it starts here and ends here. And this is our polypeptide amino acid sequence. If you can see that, it's just these different aminos that match up with each of these codons because they separated them into 3. And then we're asked what would happen if this G was deleted. So if this G was deleted, it would shift everything over a little bit. And so you'd have NF-met, methionine, and then so on and so forth. You'd keep going because that G is deleted, and so now these 3 make a sequence, and these 3, and these 3, and these 3, and these 3, and so on and so forth. And it keeps going because this one is no longer the stop codon, and then it just stops at the end of this. And then what would happen if the 3 bold, if these 3 were deleted, then it would still be the same sequence, just without this amino. And then what would happen if the amino acid sequence, what would happen to the amino acid sequence if the base in italics is changed to an A? So if this is changed to an A, then it stops right there because UAA is a stop codon. And so it would be this right here. It would be these amino acids, and then it would terminate elongation right there. So it would just be these because that changes to an A, making it a stop codon. Okay, 4. What is meant by the term a charged tRNA? So charged tRNA, you have your tRNA. It's kind of, this isn't a very good representation, but it's kind of like this. A charged tRNA will have the amino acid sequence on it. And that's all that means. How many ATP molecules are consumed to charge a tRNA molecule? So in order to bring this amino acid to the tRNA, it requires not only amino acyl tRNA synthetase, which is an enzyme, but it also requires one ATP molecule in order for that to bind. The initiation step of translation incorporates one amino acid into polypeptide chain. How many GTP molecules are consumed during the initiation step? So during the initiation step, there's two GTP molecules. One is used by IF3 and one is used by IF2, those initiation factors, in order to help the mRNA come into the ribosome, bind to the 16S rRNA subunit and the Shine-Delgarno sequence and so forth. The elongation step. The elongation step consumes two GTP molecules as well. It's one by EF2 to bind the tRNA to the ribosome. So tRNA comes to the ribosome, that's one. And then to move it to the next site, that's another GTP molecule. And then when it's terminated, only one GTP molecule is consumed by the release factor. So when that mRNA comes along the stop codon, one GTP molecule is required to terminate to the translation and breaks the polypeptide chain, and that happens. Okay, so 5. I also drew this one out. Okay. For 5, we have a wild type polypeptide chain, and then there's a mutant. So for the wild type, this is the sequence that I wrote out that could be possible, because this is the wild type amino acid chain, polypeptide chain, sorry. And then, so this is the possible sequence. And then for B, there's different mutants that occur, and you're supposed to determine what happens. So for this first one, for the first mutant, it stopped. And so we know that this could possibly have changed to an A, making translation stop right there. This one right here, we just see that one of the amino acids has changed, and it's this one. And so we see that this, sorry if you can follow my arrow, this changed to a C, making it this amino acid and not this one. And then mutant 3, right here, it's just gone all sorts of crazy. And so we know that it changed pretty early on, and then everything else was affected. So that could be like a single base being deleted. And it was this G right here got deleted. So everything got shifted over, and a new amino acid was synthesized. The mRNA that produces the wild type protein should have 9 codons. So this is asking what the functional role of the 9th codon is. So this is the 9th codon over here, and this is the stop codon. It doesn't actually add a polypeptide, or it doesn't add an amino acid to the chain. It just terminates translation. The mRNA molecule that produces the wild type protein contains an untranslated region upstream. So upstream of this, you have your 5' and untranslated region. And all that is the Shine-Delgarno sequence, which is part of the initiation step for translation to start occurring when you reach the start codon, but it doesn't start occurring here. This just helps in binding. And this is where it actually starts. So that's my video, and hopefully that helped you understand translation a little bit better and with these practice problems.